Hello everyone. Now let's study the chapter 6, Microbial Nutrition and Growth. So let's study section 1. So let's talk about the nutrition of bacteria. The nutrition for bacteria can be classified as essential nutrients. Essential nutrients means this nutrients is very important for the bacteria survive. So without these nutrients, so the bacteria cannot survive. So that means so this nutrient must be provided uh, to the living organism. Okay. So based on the molecular weight, so we can classify the nutrient as the macro nutrients. Macronutrients means this substance has a higher molecular weight. So such as this substance contains carbon, hydrogen. So they are the organic compounds. So this compounds has higher molecular weight. Another is the macro nutrient. Macro means low, so the macro means heavy. So micronutrients means this substance has lower molecular weight. So, for example, um, the mineral, so the metal elements such as magnesium, so the zinc, nickel, so the magnesium. Also, so they are also known as the trace element because the living organism doesn't need a lot of this substance, so only a little bit volume is enough. So we can also classify the nutrients as inorganic and organic. Inorganic means the substance, the substance doesn't contain the carbon and hydrogen, because the carbon and hydrogen are the elements for the organic nutrients. So if the substance contains carbon and hydrogen, so this substance is considered as organic. So the organic substance. So the good example for the inorganic nutrients could be the water, right? So the water, and so the gas like oxygen and carbon dioxide. So the oxygen, no carbon dioxide. So the metal, okay. So the magnesium sulfate and phosphate, right? So the organic nutrients contains carbon and hydrogen. So so the organic nutrients are the the macronutrients, because this kind of substance have high molecular weight. So normally they are the large polymer, okay, such as carbohydrates, lipids, proteins, nucleic acid. So let's talk about the nutrients. So the scientists have analyzed the percentage of the nutrients in the body of living organism. So they found that the water is the most abundant components. It's the most is the most abundant substance among all the components. So water account 70%, and then it's protein. So the organic compounds account 97% of the dry cell weight. So the elements like carbon, hydrogen, oxygen, nitrogen, phosphate, and sulfate account 96%. 96%. So we are made of so we are made of all these elements. So our cells can synthesize all the substances that we need. So we can get the nutrients from outside. So such as the food. So the plants and grass can use water and carbon dioxide to synthesize the food. So only a few types of nutrients need to be synthesized over 5,000 different compounds. So here is the table shows the composition of the E. coli. So we can see so the E. coli is made of all these elements, and the nutrient would be the proteins, nucleic acid. So the protein is the most abundant organic substance for the living organism. So what does the microbe eat? So based on what they eat, so we can classify the living organism as the heterotrophy. Heterotrophy means this type of living organism have to get their food from outside. Have to get the carbon and hydrogen from outside, from the organism form. That means they have to eat the other living organism to survive. The second is autotrophy. Autotrophy means the organism can make their own food. So they can use carbon dioxide and water as the material to make their food. So they can convert carbon dioxide to the organic compounds. Phototroph means so we can subdivide the autotroph to the phototroph and chemotroph. Phototroph means the organism use the carbon dioxide and water to make the organic compound. However, the energy that is used to make the organic compound is from the light. Photo means light. Okay, actually it's from the sunlight. So this is called as the photosynthesis. So, so second is the chemotrophy. That means the organism still use the carbon dioxide and water to make the organic compound. However, the energy that they use is not from light; it's from chemo. Okay, chemo means chemicals. Okay, it's from so they use so they digest other chemicals and break down the chemical bonds in that chemicals, so they can get energy from when they break down the chemical bonds. So they use this energy to make their own organic compounds with from carbon dioxide and water. So this is called chemotrophy. So here we can see these are the example for the autotrophy and heterotrophy. Okay. So autotrophy, they use sunlight as the energy source. Chemotrophy use the other compounds. Then they can build their own. They can build their own organic compounds. So they can also use inorganic compounds to provide the energy. So the heterotrophy, so we have the chemoheterotrophy. So the energy source would be from other organisms. For example, the protozoa, fungi. We also have photoheterotropy. And chemohetero sapro means the living organism will get their food from the dead body of the animals or dead body of the plants. Parasites means the living organism have to invade the host to get the nutrients from the host. Now let's talk about their energy sources. So the photoautotrophy. Photoautotrophy is also known as the photosynthetic. Okay, so the living organism will make the organic molecules using carbon dioxide and water. However, so the making those products need the energy, right? So this energy is provided by the provided by the sunlight. So chemoautotrophy actually is organic compounds for energy and to make their own organic to make their own organic compounds. They can also digest inorganic compounds. So this is called lethal autotrophy, such as they can digest the mineral. So they can digest the inorganic mineral and get energy from the mineral to make their own organic compounds. For example, some bacteria can digest the metal salt in the rock, so they can eat the rocks, and they use energy from the rock to build their own organic compounds. Chemoheterotrophy. So this means they need to get their own carbon and energy from organic compounds. So they have to eat other living organisms. So the good example would be the fermentation, and also through the cellular respiration. Saprobes means the living organism have to get their food from the dead body of animals or plants. So they can decompose the dead body and reuse the materials such as carbon, hydrogen, phosphate, sulfur, and recycle these elements to the soil. So the good example would be the fungi, right? Fungi. So the mushroom live on the woods. Parasites means the living organism have to get their nutrients from the other host. Okay, the other host from either cells or tissue of a living host. So most of these parasites cause disease, so they are also called as the pathogen. So they can cause damage, cause disease to the host. So the parasites can be the virus. So all the viruses need live in the host, right? So from host to the hermits, okay, hermits means the worms, okay. So we can classify the parasites to the ectoparasites. Ecto means outside, okay. That means they live outside of the body, of the host. For example, um, one type of hermits live on the surface of the skin, right? So this kind of ectoparasites. Endoparasites means the living organism has to live inside of the body, such as organs, tissues. Intracellular means they will live inside of the cell, okay? Like the malaria, okay, malaria lives inside of the red blood cells, right? Obligate parasites means those type of living organism cannot survive outside of the host. So all their lives have to be inside of the host. For example, the leprosy, bacillus, and syphilis, parasite. So they need to live inside of the host for their whole life. So here are the table for the elements. So we can take a look. So they are very important for the living organism. So now let's talk about the other nutrients. For example, the potassium. So it's very important for the protein synthesis and membrane function. Sodium important for the cell transportation, for the body fluid osmosis. Calcium is very important for the muscle contraction and also for the cell wall. Magnesium is the component for the chlorophyll, so that's pigment for the photosynthesis, right? And stabilize the membrane and the ribosome. Iron is the component for the cytochromes and hemoglobin. So it's also important for the cellular respiration, because cytochrome's function is for the cellular respiration. Zinc is for the genetic function. So let's talk about the transport mechanism. So the transport of the nutrients can be driven by different force. Okay, so the first one is diffusion force. Diffusion force means so if the substance concentration is high in the solution, so the high concentration of the substance will generate force to push the elements to the low substance concentration area. So this force come from the elements gradients, concentration gradients is called diffusion. Okay, it's called diffusion. So this concentration gradient from a higher concentration to the lower concentration. Osmosis is for the water. Okay, it's not for the substance. It's for the water. For example, if one substance has a high concentration gradient in the solution, so this high concentration substance solution will attract water from a low substance concentration area. So this attraction force for the water is called osmosis. So we can use a selective semi-permeable membrane to do this experiment to help us better understand these two concepts. So it's like this. So this is semi-permeable membrane. Okay. Semi-permeable membrane. So the red dot is the solute. So green. So the blue is the water. So we can dump the solute inside of the membrane and put this membrane sac in the water. Okay. Since the concentration of the solute inside of the sac is very high, so they will generate the however the membrane cannot allow the solute to get out of the sac because this is semi-permeable membrane. It can allow the small molecules get in or get out, such as the water molecule. It cannot allow the huge molecule like this solute to get out. So since the concentration of this solute is very high, so inside of this sac we generate attraction force for the water, so the water will get in. Okay, water will get in. Some water can get out, however, most of water will get in. So eventually, more and more water will get into the sac. So the volume of sac will be increased. So we can see this sac can get bigger and bigger. Okay, so this is osmosis. Osmosis is attracting forces for the water. So based on the osmosis, so we can divide the solution as isotonic, hypertonic, and hypertonic. Isotonic means the outside the solution osmosis is equal to the solute osmosis. So if we put these cells in this solution, so actually there is no net water come in or come out. Okay, for example, the 0.9% solution, sodium chloride solution. So this is isotonic solution. So if we put the cells in this solution, so the cell still have its normal shape and size. However, so if the solute concentration is very low, so this is a hypertonic solution. So if we put the cells in this solution, so actually since the osmosis of the solution is low, so more water will get into the cells. So eventually, so the cells will absorb more and more water and get bigger and bigger. So if the solute solution is very high, so this is called a hypertonic solution. So if we put the cells in this solution. So the cell, however, because so the cellular membrane is is one type of semi-permeable membrane, so it cannot the solute so the solute inside of the cell get out from the cell. Okay, so only water can be get in or get out. So the solution will attract, since this is high osmosis solution, so the solution will attract more water out. Okay, so eventually, so the cell will lose the water. So the water inside of cells. So eventually, so the cells will shrink. Okay, shrink. The size is smaller. So now let's talk about the transportation. We have two kind of transportation: passive and active. For the passive means so this type of transportation doesn't need energy. So the force for the transportation uh, is from the diffusion force. Okay, is from the uh, substance concentration ingredient. So if the substance concentration ingredient is very high outside of the cells, so the diffusion force will push the substance molecule through the channel on the membrane and get into the cells. So this is passive transportation. So so the channel would be the sample diffusion. So if we have the receptor, so if we have specific proteins, membrane proteins here, like receptors, so they can help the molecule get into the cells. Okay. However, this type of transportation still doesn't need energy. Okay. So the energy from the diffusion force. However, they can help the molecule get into the cells. So this is called facilitated diffusion. Okay. So in this type of diffusion can have a higher transportation speed than the sample diffusion. Then we have active transportation. Active transportation means so this type of transportation need energy. Okay, so if we have specific proteins here, so the protein actually works like a bump. So it can use energy and bump the molecule from the region low concentration, from the region low concentration to the region high concentration. Okay, because if the substance concentration is high in this side, so they have diffusion force. Diffusion force to so the direction of the diffusion force is to the low concentration, low substance concentration area, right? However, the bump can transport the substance from low concentration to high concentration, so it's reverse to the direction of the diffusion force. So this type of transportation needs the energy. Actually, the energy is provided by the ATP. Okay. So the active transportation can bump the nutrients against the diffusion ingredient diffusion direction. So it requires a specific membrane protein. So this is called as the bump. Okay, it's called because it's like the bump. So they need energy. So the example would be the transportation of nearly all the nutrients, such as monosaccharide amino acid, so the macro nutrients. Almost all the macronutrients need this type of transportation, okay? Active transport. Now let's talk about the cellular eating, okay? Cellular eating or drinking, okay? Well, so first is the endocytosis. Endocytosis means the cellular membrane enclose the substance and then make a vacuole to surround the substance, let the substance get into the cells and then digest the substance. So this is cell eating, okay? So there is another type of cellular eating. So the cell have to eat a huge substance, okay? So 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 we describe this eating in the for the amoeba or the white blood cells, because they have to eat they have to eat one, so they have to eat the other entire living organism, such as bacteria. Okay, so they have to eat the whole cells or huge whole huge matters. Okay, so this is called phagocytosis. Actually, so this means eat big things. Okay, so this is endocytosis means eat small things. Then the pintocytosis. Pintocytosis means actually, so this means cellular eating, cellular drinking. Okay, cellular drinking. So the cell can make a vacuole to to ingest a drop of liquid, okay, such as oil or the other liquid stuff, okay. So this is called pintocytosis. Now let's work on this question. Which of the following terms describes an organism that derived its energy and carbon from organic molecules? So I'll give you 5 seconds. So the correct answer is B, right? So their energy and carbon from organic molecules. So this chemo heterotrophy. So this autotrophy actually is use inorganic material. Photo. So this is not because autotrophy they use so they use carbon dioxide. So they don't get the carbon from the organic molecules. So actually this is heterotrophy, it's not autotrophy. Now let's work on the section 2. Let's talk about the temperature. So the temperature, because the living organism need a certain temperature for surviving. So based on the range of temperature, so we can set up this concept. So this is the minimum temperature. Minimum temperature means the lowest temperature that is required for the living organism to survive. Okay, so below this temperature, so the living organism their stop thriving, or maybe they can die. So the maximum temperature, so this is the highest temperature that is required for the living organism. So beyond this temperature, so the protein maybe can be denatured, so the living organism can die. Okay, optimal temperature means this is ideal temperature for the living organism to grow and thrive. Okay, so at this temperature, so the metabolism rate can reach to the can reach to the highest speed. Okay, so the living organism can grow fast, can grow fast. So based on the temperature, we can classify the living organism to the cycrophiles. Cycrophiles means this type of living organism can live in the low temperature. So the optimal temperature is below 50 degree Celsius. So they can also live at 0 degree. Okay. However, the maximum temperature is 20 degree. So they cannot live above 20. So this type of living organism can live and grow at a refrigerator. Okay, so they can live in the cold areas such as polar area and the high mountains. Okay, deep oceans. So they can be the bacteria, fungi, algae, and so forth. So most of them cannot cause disease for human. Okay, because they need a low temperature. So our human and many animals have the higher body temperature. So if those living organisms invade our body, so our temperature can kill this type of living organism. So they cannot cause disease to us. And that is the psychotrophy. Okay, psychotrophy. Psychotrophy can grow between 15 degree to 30 degree. Okay. Example would be the Staphylococcus aureus and Listeria monocytogenes. Okay, so they can grow at cold environments such as the refrigerator. Okay, so they can generate a toxin, and then so if the food is contaminated by this bacteria, so the food will get the toxin that secreted from this bacteria. So if the people eat those food, so it can cause the food-borne illnesses such as diarrhea, nausea, vomit, okay, and fever, and so forth. So third is the mesophiles. Mesophiles can grow from 20 degree to 40 degree. So most of these living organisms can cause disease to animals and humans. So many of these bacteria or organisms can invade animals and human and plants and cause disease. For example, the human pathogens, so their optimal temperature is 30 degree to 40 degree. And then, so the fourth one is the thermoduric microbe. So they can live at a higher temperature than the 40 degree. Okay, so this type of bacteria can live even during the pasteurized technique. Uh, so they can survive even uh, so even so they can survive even after so even after steroids technology such as the pasteurization. So this is uh, pasteurization means we heat the food at 30 degree. Okay, 30 degree. So they can survive at 30 degrees. So they can still survive after pasteurization. A good example would be heat-resistant endospore. Okay, endospores from Bacillus and Clostridium. So the thermospheres. So the optimal temperature is over 45 degree. Okay, so they can live in the soil, water, uh, mm, associated with the associated with the volcano and compost piles. Okay, all the some surface of material face to the sun. Okay, since they can survive in such high temperatures. Uh, so some living organisms can survive even higher 45 degrees. For example, like 50, 70 until 80 degree. Okay, 80 degree is very hot. Mm. So most of these living organisms are the bacteria because eukaryotic cell cannot most eukaryotic cannot survive over 60 degree. Some extreme thermospheres can survive between 80 and 121. Now, so the gases. So, so based on the gases that the living organism need, so we can classify the living organism uh, based on if they need the oxygen, okay, or the carbon dioxide. Now let's talk about the oxygen. So many living organisms use oxygen uh, to to di- to burn the sugar to burn the carbohydrates to provide the energy. However, uh, so when the oxygen burns the carbohydrates, so the oxygen can also make the so the oxygen can also make the oxidative stress to the living organism. So this oxidative stress is considered as is considered as the oxygen toxic for the living organism. So this type of toxic can be fatal. Okay, it can kill the living organism. It can kill the living organism. Okay, so if the living organism micro need to use oxygen to for the cellular respiration, so they have to have their own enzyme to detoxify the oxygen. Okay, so if they can use oxygen and can detoxify oxygen, so we can classify this bacteria as aerobe. Okay, and then so second class would be the the microbe doesn't use oxygen and doesn't have the enzyme to deoxify oxygen. Okay, so third group would be don't use the oxygen, but they have the enzyme to deoxify oxygen. That means they can survive in the in the in the environment that contains oxygen. So why oxygen can generate the toxic to the living organism? Because oxygen can transform to several toxic products. Okay, the first is singlet oxygen. So all this is called free radicals. Okay, free radicals. So they can destroy the cellular membrane. Okay. So second is superoxide ion. It's highly reactive. Okay. So third is the hydrogen peroxide. Hydrogen peroxide is toxic. So we can buy this solution from Walmart or CVS, right? So the first is hydroxy radical. So most of the cells have the enzyme to deoxify these oxidative products. For example, the superoxide superoxide and superoxide dismutase. Okay, so this is also called as SOD. So SOD can convert the superoxide ion to the hydrogen peroxide. Okay, and then so the second enzyme would be the catalyst to convert the hydrogen peroxide to the water and the oxygen. So water and oxygen. So water doesn't have the toxic oxygen actually. If so, oxygen itself doesn't have the toxic. However, if they transfer to the free radicals, so they have the toxic. So if the living organism can use oxygen and can detoxify oxygen, so they are called as aerobes. Okay, aerobes, they need the oxygen. So if they cannot survive without oxygen, is called obligate aerobe. Okay. So the example would be some fungi, protozoa, and bacteria. Okay, microaerophiles. So this type of microbe need a little bit of oxygen. Okay, however, without the high concentration oxygen, so they can still survive. So the good example would be the Helicobacteria pylori. So this type of bacteria live in the stomach. Okay, so inside of the stomach, so the oxygen concentration is very low. Okay, so this type of bacteria can survive in this condition. Facultative anaerobes. Okay. So this type of bacteria actually doesn't need oxygen. Okay, doesn't need oxygen, but they can still survive. So they can still survive if the oxygen presents. Okay. So for example, many gram-negative intestine bacteria and staphylococcus. Okay. Anaerobies. Anaerobies means so this type of microbe doesn't have the so their metabolism doesn't need the oxygen. Okay, so they also don't have the enzyme to detoxify oxygen. So if they cannot survive in the environment with the oxygen, so this is called obligate anaeroby. Okay, because they cannot detoxify oxygen, so they will be killed by the oxygen because oxygen actually can generate the free radicals. So interestingly, many of obligate anaeroby exist our oral in our mouth. Okay, in our oral cavity. Okay, even when we speak, so when we speak, so the oxygen get into our mouth. So however, in our mouth, so there are many obligate anaeroby. Okay. Also in the GI tract. Okay, intestinal bacteria. So most of them are the obligate anaeroby. Aerotolerant anaeroby. So still don't need the oxygen for their metabolism. However, they can survive and grow in they can survive and grow if a certain concentration of oxygen presents. Okay. For example, the lactobacilli. So we use lactobacilli to make the yogurts. Also, we can buy the so the pro so the so we use the lactobacilli to make the yogurts and all kinds of pickles. Also some kind of streptococci. Yeah. Now let's talk about the carbon dioxide. So the bacteria need a high concentration oxygen. Need a high concentration carbon dioxide is called capnophils. Capnophils. For example, the Streptococcus pneumonia and Brucella and Neisseria gonorrhea. Because the Streptococcus pneumonia, this type of bacteria lived in the bronchi and lungs. So so in those organs, so the carbon dioxide concentration is very high. So these bacteria actually they need a high concentration carbon dioxide to survive. So we can use this characteristic to isolate those type of bacteria. Okay, so we can inoculate the specimen to the medium and culture this media in the high concentration carbon dioxide. So the other microbe will die, will killed by the high concentration carbon dioxide. Only those bacteria can survive. So we can use these characteristics to isolate capnophil. So then let's talk about the pH. pH is to measure the acidity and alkalinity. So the range of pH is from 0 to 14. Okay, below 7 is the acidity. So above 7 until 14 is alkalinity. Okay, 7 is the neutral state of pH. So majority of organisms live in a neutral range. Okay, from 6 to 8. So this is the neutral range. However, there are some living organisms can survive in acidic environments. We call this as acidophils. Acidophils. For example, Anglena mutabili, so mutabilis, okay, and Thermoplasma, Picrophilus. So if the microbe can survive in the alkaline condition, this is called alkalinophils. Alkalinophils like nitronomas and protests. So protests can create alkalinous products to make the solution to make alkaline solution. For example, they can make the urine alkaline, okay, because so that will provide an ideal environment for their survival. So some type of microorganism can live in high solute concentration solution. So this means they can survive in the high osmotic solution. So for the high concentration of salt, so if the microbe can live in the high concentration salt solution, this is called halophils. So the obligate halophils would be the halobacterium and the halococcus. So they can live in the 25% salt solution. Facultative halophils, so they can survive in the high salt concentration solution. So they can still live in the normal solution. Okay, like Staphylococcus aureus, so they can live from 0.1% and to 20%. So this is facultative halophil. Radiation. So the some type of light can generate because the light has energy, right? So some type of light can generate the toxic oxygen products inside of the living organism. So this type of toxic products can kill the microbes. Okay. So some microbes can produce pigments such as carotenoid pigments to attenuate the toxic oxygen products. Okay. So in clinic, in hospital, so they use the ultraviolet light to to kill the microbes. Okay, to kill the microbes for the hygiene and steroids, right? Now pressure. So if the microbe can survive in the environment with high pressure, for example, 1,000 times higher pressure than 1,000 times of the atmosphere pressure. Okay. For example, in the deep ocean, so they can survive in deep ocean because deep ocean has high pressure. Okay. However, those type of bacteria can only live in that environment because if those type of bacteria return to the surface of ocean, so they will die because their structures are adapted to the high pressure environment. Okay, if the pressure is gone, so their body will be ruptured, will be expanded and ruptured. So actually, so the living organism doesn't live alone. Okay, so they live with other living organisms, such as so the microbe live with other microbes. Okay, also live with other multicellular organisms, such as animals and plants. Okay, so the associated living could be beneficial, harmful, or no effect. Okay, so the interaction could be the obligatory or non-obligatory. So some microbes have to live with the other microbes. Okay, so based on the relationship, so we can classify the bacteria living organism as the symbiosis. Okay, symbiosis. Sym means together. Okay, sym means together. Biosis means the living organism. Okay, bio means living organism. Sym means member of symbiosis. Okay, so they are so these are the main type of symbiosis. Um, mutualism. Mutualism means so the organism live together can be beneficial to each other. Beneficial to each other. Commensalism means so the organisms live together. However, they don't have effect on each other. So neither harmed or neither benefits. Okay. Parasitism means one living organism get a benefit and another living organism will be harmed. Okay. Antagonism. Antagonism means actually the living organism don't live together. Okay. For example, antibiosis. Okay, antibiosis. So one living organism secretes the toxic chemicals to the other living organism to kill the other living organism, and to gain more space and more nutrients from the environment. Okay. So this phenomenon is very common in the soil. Okay. Uh, so that means this is a competitive relationship between the so among the living organisms. So they uh, so they kill the other living organisms to gain the space and nutrients. Synergism. Synergism means so the partners can get the benefit from each other. However, this is not necessary. Okay, this is not necessary. So a good example would be the gum disease. Okay, so the dental cavities. So and some bloodstream infections. Okay, so let's talk about the dental cavities. Because for the dental cavities, so this is a good example for the synergy. Okay, so the pioneer microbes were attached to the surface of the dent, okay, surface of the teeth. And then, so this pioneer microbe will secrete the polysaccharide materials. So this is called bare films. And then, so this bare film will be secreted more and more, and to make a layer of film on the surface of teeth. So this layer of film is like a house for these microbes. So so the microbes can stay inside of this film, and so this film can protect the microbes. Also, the microbes can get the nutrients from the films. Also, the other microbes can attach it to the films and get in the films, live together with the pioneer microbes. So this is the example for the synergy. Okay, so so all the microbes can get a benefit from the biofilm. However, this is not obligated condition for their survival, for they attached to the surface of the teeth. So now let's work on this question. Which of the following describes an association between microbes in which one organism is benefited and one is harmed in some way? So I give you 5 seconds. Okay, so the correct answer is parasitism. Okay, parasitism. Because one get benefit, another get harmed. Okay. Now let's work on section 3. It's about the cell division. So since the cell grows and reproduce, so they make more and more daughter cells. So how does this happen? Because one cell divides, so first of all, so the microtube will contract and to form a binary fissure in the midline of the bacterium. Okay, before the microtube the contraction, so all the organelle had to be duplicated, and the chromosome is duplicated. So they have 2 copies of their chromosome and organelles. So so the microtube will contract more and more. So the fissure, so the binary fissure will be deeper and deeper, and eventually divided the microbe to 2 daughter cells. So the two copy of organelles and the chromosome will be distributed equally to the 2 daughter cells. So the one cell will become two cells. Okay, it's like this diagram. Okay, so first of all, so that's the duplication of the organelles and the chromosome. So we have 2 copies, and then so this is a binary fissure. Okay, binary fissure here. So this is caused by the contraction of the microtubules, and then so the fissure contacted together to divide the cells to 2 daughter cells. So the time required for complete the fission cycle from parent cell to daughter cell is called generation time. Okay. because so from one parent cells to two daughter cells, so this is one generation. Okay, since the number is doubled, is from 1 to 2, so generation means increased population by factor of 2. Okay, 2 is 2. So if the environment condition is optimal, so the microbes will increase their numbers at a constant rate. So this constant rate is based on the generation time. Generation time is fixed. So for some living organisms, so the generation time is 30 to 30 minutes. Okay. So the shortest is 10 minutes, 12 minutes. Some need more time, 10 to 30 days. Okay, like a Mycobacterium leprae. Okay. So this is diagram shows growth. Okay, actually, so the number will be doubled. Okay, doubled. So we use this equation to calculate the increased numbers. Okay, so this is n means number. Okay, so, microbes number. T means the time points. Okay, n means the starting numbers. So the two is variable number because because the generation is so the generation rate is doubled, right? It's double, so that means 2. So n, so the exponent n means the generation time. Okay, generation number. So we can use this equation to calculate the increased population. Now let's talk about the growth curve. Okay, growth curve. So growth curve, how do we measure the growth curve? So for example, if we culture a microbe in a tube, for example, the volume of the broth is about 500 milliliter. So we can inoculate 1 milliliter every for example every 60 minutes. Okay, and then we capture the plates, and then we can count the colonies. Okay, it's like this diagram. Okay, so this is the interval time is 60 minutes. Okay, so for example, this is 5 milliliter. We can inoculate for example 1 milliliter on the plates, and then so later on, so we can see because so the microbe will grow more and more in this picture. Okay, so later on, so we will get more and more microbes population. So in the beginning, so this is lag phase. Lag phase is flat curve. Okay, flat curve. That means the microbe number doesn't increase. So the reason for the lag phase is that either the microbes doesn't start the growth yet. Okay, or the cells are too few. Okay, are diluted too much. Okay, so they need time to reach a certain numbers. Okay, and then is the exponential growth. So this is logarithmic phase. Okay, so the growth curve will increase dramatically. Okay. So the reason for this phase is that so the microbes have enough nutrients in the mediums. Okay. Also, the condition is optimal, so they have the optimal temperature, and so they will grow fast. Okay, grows quickly. And then so this stationary phase. Stationary phase means uh, so once the cell grows, so some cell will die, right? So the cell microbe cannot survive forever. So some will die. Okay. Also, when the microbes grows in the medium, so the microbes need eat and drink. Also, they need pee and pop, right? And so need deposit the waste to the medium. So the waste will be more and more. So high concentration of the waste will kill the microbes. Okay. Also, since more and more microbes eat and drinking, so the nutrition is less and less. So this condition will limit the growth of the microbes. So so that means more microbes will die. Okay, once the births and dies are equal, so this is a stationary growth phase. So the curve is flat again, and then is the death phase. Okay, death phase, because the media condition is worse and worse because there are more waste than the nutrients. Okay, so in this phase, so the microbes dies is more than microbes birth. Okay, so that means so the survived microbes number decrease. And then so this another concept is variable non-cultured state. Okay, so many cells in a culture in the death phase still alive, but are dominant. Okay, so we can see it's like this diagram. Okay, so this is the lag phase. So the microbe doesn't start growth, and then so this is exponential growth phase, and then this is stationary phase. So the microbe birth is equal microbe dies, and then so the death phase. So death is more than the birth. So so a good example for this curve is that for example, we get so the people are sick. Okay, so if we get infections, so eventually, so if the microbe doesn't grow, so it's like so it's lag phase, so it will not cause the disease. It will not cause the symptoms. However, if they start growth, so this exponential phase, so they will cause, so since more and more microbes are, are reproduced, so they will so they will release more and more waste, release more and more toxin to cause the symptoms, cause people sick. Okay. Uh, Also, so in these phases, so if we administer the antibiotics, so it's very easy to use antibiotics to cure the microbes in that phase, because in that phase, so the microbes grows quickly, so they need to eat and drink a lot. Okay, so if we give the medication to the patient, so the microbes can eat more antibiotics inside, so the antibiotic can kill more and can kill more microbes. So so the microbes are more vulnerably in this condition to the antibiotics. Okay. Also, so the people under infections will be more contagious at this phase, right? Because the more microbes are generated, so they can be spread to the other people. So we can we can also analyze the microbes during different phases. Okay, once the microbe grows more and more, okay, so they will make the solution cloudy. Okay, so this is called turbid. Okay, turbidity. So we can measure the degree of turbidity to determine the number of so the population of microbes. For example, for example, if this is the lag phase, so there are very few microbes, so the medium is still clear, transparent. So if we use this device, so it cannot more light to get through. So if we use this device to measure the density of light, so we get a high number of density light. So if, for example, in the expansion phase, so there are more microbes, so the solution, so the medium is cloudy, is turbid. Okay, so it cannot too much light get through. So if we read the light density, so we get a low density. However, that represents the higher population of microbes. So we can count the microbes number by direct count. Okay, with glass plates. Also, we also can use the machine to count. So this is called counter. Okay, we can also use the flow cytometry. So we stain the microbes with fluorescent, and then so we let the machine to count the fluorescent signals. We can also use real-time PCR to count the microbes population, because higher population of microbe means there are more microbes DNA, right? Because one microbe has one set of DNA. So we can run the PCR. So if there are more microbe DNA, so we can get because the PCR is measures the thermal cycle number, right? So if there are more DNA, that means we can get the very low thermal cycle number, right? For example, we can get 10, okay, instead of 30, okay? That means, so if we get 10, that means there are many microbes. So this is direct count, okay? So we use these glass slides to measure the microbe. We visualize the microbe with the microscope, and then we count the microscope. We count the microbes, okay, in this field, and then so we can know the population of the microbes. So this is the counter, okay. So we drop the solution of media. So once the medium flows through this tiny tube, okay, so the detectors can count the microbes through this tube. Now let's work on this question. Put the steps of bacteria growth curve in correct order. So I'll give you 5 seconds. Okay, so first is the lag phase, right? Lag phase. So the second is the exponential phase. The third is the stationary phase. The last one is death phase. Okay, so this is appendix, so you guys can use this for your reviewing the contents of this chapter.